was actually here 20 years ago, but I can't quite remember whether I gave a talk. Before I start, I'm a little bit wobbly today. Usually when I give a talk, if I'm wobbly, it's because of the grappa that I consumed the evening before and combination of that lack of sleep. And today it turns out to be that I have something which goes by the beautiful name of labyrinthitis. So if I sort of am a bit unsteady, that's why, not because I had too much to drink. Anyway, so it's absolutely wonderful to be back here 20 years later, a bit sobering, 20 years. And I can't remember too much about the previous conference. Certainly can't remember whether I gave a talk. Apparently I can't remember whether I bought a present for Pete Hudson from Karen Day. Can't remember any of these things. Anyway, however, if I did give a talk, what I would have been banging on about at the time with Karen Day would be whether the epidemiological features of Plasmodium falciparum, which is a malaria parasite, can be explained by assuming that this parasite population actually exists as a set of antigenically discrete strains. So the fact that malaria falciparum was diverse had been recognized for years. 20 years ago, the molecular methods were just becoming available to dissect this diversity. But what Karen and I were proposing at the time was that actually this diversity was organized in such a way that the strains behaved so as that malaria could actually be conceived of as a kind of collection of strains that were independently transmitted, antigenically discrete. We said that several features of malaria sort of fell into place when you made this assumption. That's all we were going on at the time. And one example of a particular epidemiological feature that could be explained by assuming that falciparum was a construct of discrete strains was the fact that the clinical disease in malaria is very diverse, but severe malarial disease takes two quite, again, discrete forms. Children in sub-Saharan Africa tend to die either, of severe malarial anemia, which is a profound anemia, or of cerebral malaria, which involves the vasculature of the brain being disturbed by accumulation of infected parasites. And these two syndromes are very, very different in their clinical features, and they also have very distinctive age distributions. So this is the type of thing that we were saying, take these and other features together, you can explain all of this. It's like putting a jigsaw puzzle together. If malaria is a set of discrete strains, each with its own sort of virulence characteristic and whatnot, then you can explain something like this really very, very parsimoniously. This, of course, caused the malariologists all to jump on us and say, how can that be true? And of course, Chris here was certainly among the skeptics and possibly remains so. The main reason for this, I'm not quite sure why Chris was so skeptical, but but the main reason why the parasitologists remained unconvinced or could not quite understand how this could be the case is because malaria has this incredibly complicated life cycle with several different stages to its life cycle, and within the human host, each of these stages, several antigens are presented at each of these stages. Many of these antigens are polymorphic, and then of course in the mosquito it has sex, so all these genes, different polymorphic genes, get scrambled up. So really it was rather difficult at that point to reconcile these two ideas, that you had antigenically distinct strains and this recombining pathogen population with enormous diversity at every level. We tried at one point to just say, well, maybe it's all down to one dominant antigen, and I'll come back to that dominant antigen later, but it turned out that that dominant antigen itself was encoded by a multigene family, and each parasite carried over 60 copies of that dominant antigen. So there was no way we were going to get out of this. So so I realized that I was going to have to try and look at this problem by developing models that captured this diversity, this multi-locus diversity. And so I started to build a multi-locus model which I personally have been using since then to explain pretty much everything, for which I do not apologize. But I'd like to present it to you as an example of how one might go about looking at these sorts of systems. Obviously, there are many cleverer ways of doing it, some of which I hopefully will be able to touch on if I don't fall over by then. So within this, to build any kind of multi-strain model, as Julia has mentioned, you need to think of tricks, otherwise you just end up with this huge mass of equations. And this is still actually, of course, a huge mass of equations, but a trick that I've used here is to consider, first of all, to describe the strains of a pathogen in this kind of multi-locus framework where you have a set of loci and, if you like, whatever, alleles, epitopes, variants at each locus. So that just gives you a set of multi-locus genotypes. And then what I assume for each genotype, and I don't very often get a chance to show equations, so I'm going to waste a bit of time on them. I assume that that the compartments that describe the proportion of people immune to each genotype can overlap. For each compartment, you can write down a very simple equation just saying that the proportion that's not immune to that genotype gets infected by that genotype, some lambda I, force of infection I, and then dies at some rate. That's very straightforward. And originally, actually, the trick I tried was one that Adam Kucharski has now very successfully used in an age-structured model, which is just to assume that these overlap in a random fashion. This is, of course, not correct when you don't have age structure. And with the help of Neil Ferguson, I was able to actually correct this. And what you can't make that assumption that they overlap randomly. And so you need an extra variable which describes the proportion of people that occupy a compartment which includes everyone who is immune to a particular strain type, let's say AX in this example, but also everyone else who's immune to anything that's related to AX. So in a simple two-locus system where you've got A and B at one locus and X and Y at the other, it would be just AY and BX. And once you've got these two specified, the only thing that remains to be done is to write down an equation for the proportion that's infectious for strain I, and essentially what you say is, well, anyone who's outside of this, with WAX, which is anyone who's been infected with anything related to AX, can, of course, simply be infected, and people who are within the compartment that includes people who've been infected by something related to AX but not AX itself have a probability 1 minus gamma of becoming infected, and then they recover at a rate sigma. And this gamma parameter really tells you what it stands for is the strength of immune selection. So if gamma is very high, that means that being infected with AY completely ruins your chances of being infected with AX. In other words, the epitope-specific or variant-specific response against A is enough to prevent infection by anything carrying A. And the curious, when I set up this model, what I was hoping is I could show, depending on the rates of recombination, you can still get some structuring, blah, blah, blah. And what I actually found, which was very exciting to me, was that under strong immune selection, in other words, when gamma is high, what you get is a sort of natural structuring of the system. In other words, the pathogen population self-organizes into a subset of strains which do not share epitopes with each other. So, and this, of course, I'm showing this for a very simple two-locus, two-allele system, but believe me, I've looked at this now for almost 20 years. You can scale this up to whatever dimension you like, and you will always, as long as you have strong immune selection, you will always get a subset of strains dominating that do not share epitopes. In other words, what's going on here, of course, is just competitive exclusion, and the result of that competitive exclusion is a favoring of a subset that doesn't share any epitopes and therefore aren't competing with each other. So we published this work in 1996, but although my initial hope had been to say, this is what explains malaria diversity and the circulation of antigenic types in malaria. In fact, we'd only just in 1995, not we, other clever people, had been able to clone the relevant gene, the VAR gene, that we'd focused on as being the primary antigen that was responsible for strain structuring in malaria. And to date, I think that the jury is out on whether malaria is actually structured in the way that this model suggests. However, luckily, I met at the time a young chap by the name of Martin Maiden, who had a lot of data on exactly what I wanted, combinations of alleles, if you like, variable regions on a surface protein of Neisseria meningitidis, a bacterium that causes meningitis. And these are some data collected in England and Wales from disease cases of meningitis, about 220 samples. And if you see, Martin had been sequencing these two epitopes, and what was clear in his data is that these combinations are not only non-random, but also non-overlapping, which is what the model predicts. So since then, Martin and I have been collaborating very closely. He, in fact, moved to Oxford a few years after we published the paper. And, I mean, this is, I think, an important point that should come out, that Pete tried to bring out. This is not truth. This is not a model saying, yes, I've solved the problem. This is a hypothesis. In 1996, or whenever we published this, this was a hypothesis. There are several people who, again, even the meningitis community and others, who also jumped on this and said, these are just disease isolates, these two things are too close together. Yes, of course, that's absolutely true. And since then, we've been working to address those issues. We now have much larger data sets. Obviously, we now have whole genome sequencing. So we can see these patterns still in very large data sets. We've got longitudinal data sets from the Czech Republic, which also show these patterns. We've looked not only at this particular combination of epitopes, which are in fact quite close together, although being close together does not make them, they can guarantee some degree of non-randomness, but not non-overlappingness. But we've looked at other things that are much further away, so couldn't possibly linked. And this pattern is always recovered from these data sets. One of the interesting directions that this has also taken, and one of the mysteries about all of this non-overlappingness, was how it actually arose given the different levels of diversity at these different loci. So if you have a symmetric model, you can get at these lovely non-overlapping subsets. But as soon as you have some asymmetry in the numbers of alleles, the thing becomes a lot more messy. So this is a realization with 3 loci and 3 alleles. 3 loci with, I can't remember exactly how many alleles at each locus, but different numbers of alleles at each locus. I can't read this. And in a deterministic solution, you get actually not a very clean signature of non-overlappingness. But Caroline Bucky, who during her PhD with myself and Martin Maiden, set up a stochastic implementation of this model. And what she found was that once you have set it up, use a stochastic individual-based method to look at this problem, that you actually get some of this asymmetry disappearing. Some of these strains simply drop out, and the whole system collapses back down to a nice symmetric state. So I think this is an example, and I've put this up especially for Andrew Barber, where the stochastic model showed us something different to what we had in the deterministic model, and actually cleared up this mystery. And this is also borne out by data that's been collected in several countries since then, where you see a lot of diversity in difference, asymmetry in the actual diversity in these 3 loci. These are, again, the 2 that Martin and I looked at, plus another called the FET-A. But you see that at the population level, this all falls out, and you get this kind of symmetric pattern emerging. We've also been using this model not just to look at genes under selection, but genes that are not under selection, because Martin, in fact, pioneered the use of typing housekeeping genes to look at the evolution of Martin and other people. He would hate to take the whole credit himself. To look at housekeeping genes to type bacteria. So we've been developing models that include not just the variable antigenic determinants, but the housekeeping genes, because it turns out that the housekeeping genes actually have a lot of structure. It's not non-overlapping, but there is a lot of LD. And again, Caroline Buckey was able to show, using a model that combined the antigenic determinants with the housekeeping genes, that you can explain, using this kind of a framework, why the housekeeping genes might also exhibit this high level of structure, which had been previously attributed basically to processes of descent. So the original idea was that bacteria are clonal, and that's why they have these housekeeping gene lineages that are just described by these housekeeping genes. And so our new idea is, no, that's not what's going on. Actually, certain combinations of housekeeping genes are actually slightly more fit than others. And within this kind of a framework, with competition, strong competition occurring between housekeeping gene combinations by virtue of sharing antigenic determinants, you can actually also get this lineage structure. And this sort of non-overlapping association actually between antigenic, so another feature of this, so you get the lineages, but you also get the lineages associating in a non-overlapping pattern with the antigenic types. And this is something that is not just characteristic of Neisseria meningitidis, but really of various other important bacterial pathogens. And we've been using this, Ellen Watkins, a graduate student in our group, has been trying to use this model to look at pneumococcus. This is some recent data from Croucher et al., Mark Lipsich's group, showing the different antigenic types, the capsular serotypes of pneumococcus, this whole genome sequencing data showing how, if you like, their metabolic types segregate with the antigenic types. And like many pathogens, many other bacterial pathogens, it does have that characteristic feature of antigenic types and clonal complexes or metabolic types, or whatever you want to call them, exhibiting unique associations. And what Ellie has done is set up models to look at what happens then when you disturb this kind of a system. So not the standard vaccine sort of strain replacement model, but what happens when you take away some of the antigenic types in this kind of a modular system where different antigens can associate with different metabolic types. And what that shows is that actually you can get metabolic types hopping onto new antigenic types, which is something that is evident in the data. This data from Angela Brueggemann, where, you can see that 19A, which is not a strain that's included in the vaccine against pneumococcus, has acquired some clonal complexes that were previously associated with 19F and serotype 4, which are strains that are in the vaccine. And what we predict, what this model shows is that so what this means is that non-vaccine serotypes can actually acquire the metabolic types of vaccine serotypes, which could obviously lead to an increase in their transmissibility and indeed also in their virulence. So that's one route that we've taken with this, but another behavior of this model, which once I'd fixed the model with help of with Neil Ferguson's help, one of the other types of behavior that we found from this model is that you can get sequential, rather than a standing sort of crop of antigenically distinct strains, you can get a sequential dominance of antigenic types. So you can get one type following the other. And in the last 10 years or so, I've been working with Maria Recker and Ollie and others to see whether these principles can actually also explain the population dynamics of influenza. Where this differs from the conventional antigenic drift model is that we believe that the way this model works is through the flu virus having some epitopes at least which are not anywhere near as which are not very variable, let's put it that way. So the conventional idea is that flu has these epitopes which keep changing and sort of continually accruing change, and you have this sort of linear increase in antigenic distance with time. In our model, our model is driven by epitopes which have limited variability, and what the flu virus is trying to do always is come up with a combination of these limited epitopes that guarantees that it will succeed in a particular immune landscape. How does this square with... So what this does, what we like about our model is it allows the sort of successive emergence of antigenic types in a manner that is relatively independent of the mode and tempo of mutation, because it's the landscape of shifting host immunity that determines when a new strain emerges, rather than the mutational capabilities of the virus. I mean, it's not totally actually independent of the mode. I think we're a bit rash in saying that. In recent work by Daniel Zinder and Trevor Bedford have shown that indeed for this model to actually produce the trees, the real flu trees, some dependence on mutation is almost definitely necessary. How does this actually square with, VMAP? How do we actually, how does this map square with this idea? Well, what we would say is that what this map, that there are on the hemagglutinin molecule several epitopes. Some of these are of high variability, and we believe that the hemagglutination inhibition assay, which has been used to generate many of these maps, is possibly focusing on these epitopes of high variability, which perhaps are near the receptor binding site and play a role in the binding of the virus to, well, in this case, RBC. But we think that hidden here are shared epitopes on this on hemagglutinin, which are of intermediate variability. They're not the kind of broadly the epitopes that are very low variability, which elicit broadly neutralizing antibodies that people are hoping to identify and finding difficult because they aren't very immunogenic. We're saying that there are epitopes that are immunogenic but have limited variability, and at the moment what I'm trying to do is hunt for these epitopes in collaboration with people who can actually do those killer experiments that are absolutely necessary to nail this. Obviously it makes a difference. It will make a difference if they're there or not. So epitopes, so this is kind of what this model actually, at the end of the day, does have embedded within it is the notion that these epitopes actually do differ in their degree of variability. Now this is something that's been recognized by a number of different models, and for example Martin Novak's famous HIV model, but just simply by virtue of having two different immune responses against the HIV virus. One way at least of interpreting that was that the virus contains some epitopes of high variability which are specific and other shared invariant epitopes. And indeed we've actually recently been trying to develop a new model for HIV where we assume not only do these things, do we have epitopes that differ in their variability, but also in their quality and duration. And I won't, in the interest of time, go into the details of that, but we are trying very hard right now to publish a paper that, based on that idea, that suggests that the breakdown of HIV control is primarily due to the loss of antibody induction rather than the loss of the failure of the CTL response, which is the dogma. And this is work done with Paul Vikram Ratna, Paul Klenneman, and again, Ollie. Ollie is like a Zebedee in this session, for those of you who know. Now, what we're using in this model is actually an idea that was borrowed from earlier work done with Mario Recker and Chris Newbold to explain, and now I'm coming to the within-host dynamics part of all of this, to explain the within-host dynamics of malaria. the same genes that I've been talking about in the context of the population level diversity of malaria. These same genes, I mentioned there were 60 copies of them. Why are there 60 copies of them? It's because the pathogen, the parasite uses them to evade immunity during the course of a single infection. So the parasite plays these cards one at a time to prolong its infection. So that's all very well. Again, at a descriptive level, that sounds great. You know, lots of cards, play one card at a time, and you get away with a longer infection. Some of the neurosyphilitic studies that Pete mentioned, very clear examples of that. But of course, there's a big problem here. What we really have is a population of parasites. In order to make this a successful strategy, they have to all play the same card at the same time. Otherwise, it doesn't work. So how that might occur, how this orchestration might happen, is something that Mario Recca published a model on, again, a sort of with a multi-locus framework, again, with some epitopes that were highly variable and specific to each variant, and other epitopes that were shared between variants. And again, these shared epitopes have the same effect as the shared epitopes in flu in restricting diversity within one particular peak, which is in fact of benefit to the pathogen because it allows it to conserve its repertoire. So the host immune response in this case is actually helping the pathogen orchestrate its this process. So now we're back again. So this particular gene that I'm talking about, we're back again to malaria, but we're now talking at the within host level about this gene which, as I said, encodes, of which there are 60 copies, and this gene encodes a protein called PFEMP1, which is involved in cytoadherence, which is the sticking of this parasite to the endothelium, which is a process that's involved in virulence, hence the link again to the disease syndromes. So cerebral malaria patients more likely to have a type of gene that sticks to a receptor that's prevalent in the brain, that kind of thing. So what we have here is now a system where we've got the within host and the between host dynamics. Can we put them together? Some very nice work recently by Yela Sirandup and Mercedes Pascual has done just that within, again, a sort of individual-based model. And again, I don't have time to talk about it, but they've been looking at how, where you have a situation where you are looking at an individual host who now receives this parasite, which has a repertoire of variants which are going to be expressed sequentially, and so you won't necessarily acquire immunity to that whole repertoire. How does that alter the dynamics at a population level? And they have some very interesting results. Again, they find a lot of structure, but that structure, of course, depends on the transmission intensity and the rate at which these things are expressed, and a whole variety of things that very interesting. Elaborate, I'll bring a little bit more sort of finessing on this idea, but expanding it out and also making it more amenable to testing, because now we have a lot of this data on the VAR genes emerging. So we need models like this to be able to look at the relationship between these data and these ideas. And finally, also another very nice paper from Caroline Bucky and Mario Recca, looking at the finer structure of this incredibly complex gene family, and looking at the domain structure of these genes, using models, again, that incorporate the within host and between host modalities. So obviously, unfortunately, I don't have time to go into these, but I just wanted to basically say that I think this slide that Julia and James showed in their wonderful sort of review of what's happened in this field. I do think that indeed there is a lot of value in attempting to develop models which incorporate both of these processes from the start. This is one example, I think Sebastian's talk. There's so many ways in which this can be done. Sebastian's talk was another example of where all this data that is being generated can be, we can start to think about how to look at these things. I think, I'm very optimistic. I think the younger generation are very keen to do that, and I think things are moving just in the right direction. Thank you very much.
Any questions?
I have a question to this, to the antigenic types in these bacterial pathogens. You showed these nice associations between the antigenic types and these housekeeping genes. Now, given that you have disruptive selection on the antigenic part, it's sort of not surprising that you get these different types. But then you have these associations with housekeeping genes. So do you attribute that to epistasis between different types of housekeeping?
Yes, there's some epistasis, yes.
Do you have an insight what this epistasis is grounded in?
Yeah, so the epistasis is basically, what we're saying is certain combinations of housekeeping genes are slightly fitter than others. Now, without the antigenic selection, this wouldn't matter. You'd still get basically a mush of combinations of housekeeping genes. But once you have the antigenic selection, it's as if you've got these antigenic islands on which these things have to compete. And so the thing with the, forgive me, Pete, with the highest R naught will actually win out on these antigenic islands. But in addition to that, we also assume, what we find works very well, is if there's some level of also ecological competition between similar metabolic types. So that, let's say, if you're immune to a particular antigenic type, if you're infected with a particular strain, you're immune to that antigenic type, so you can't be infected by that antigenic type. But while you're colonized by that strain, you also have some resistance against a similar metabolic type. So a new antigenic type with a similar metabolic type also has a problem while you are colonized by that particular metabolic type. So that then creates the non-overlapping associations between metabolic type and antigenic type.
Heterozygous disadvantage for some sort.
Yes, essentially. Yeah, well, so yeah, I mean there are a number of assumptions underlying this, but it seems to be the case with many bacterial pathogens that you get very strong associations between, let's say, capsular serotype and metabolic type as defined by housekeeping genes, or now with WGS we can get much better signatures of metabolic type. Stunned silence.
Thank you to all speakers this afternoon, that was great.